And we are. And yes, and that will be our housekeeping to do, yes, before we start. Okay. So I want to welcome you. This is our fourth webinar, and we I'll be doing a brief introduction of the project in place of Dennis, our coordinator of the project. Unfortunately, he wasn't able to join us for reasons that he couldn't control, a train problem. And but I'll try to give a brief introduction here before our speakers can start. Um, the first point I want to make is in GeneBEcon, as a project, we are using gene editing to develop the virus-resistant potato with an optimized tuber starch quality, so for pesticide-free production in the field and a chemical-free industrial processing of the starch. We are also applying gene editing in the microalgae. We are producing high-value compounds while using the residual biomass as chicken feed for a zero-waste bioeconomy. And as a more general purpose tool here, to the topic of this webinar, GeneBEcon is also developing a comprehensive and versatile toolbox for gene editing containing protocols and a guided workflow, which we'll see today through an editing um, for an editing experiment, which is applicable in many different species, actually. This work is in alignment with the expectations from the European Commission. It was emphasized in the context of the project itself that this project should advance the implementation of the new genomic techniques and contribute to the Green Deal, the Circular Economy Action Plan, the bioeconomy strategy, and zero pollution bio-based solutions, right? Biotechnology solutions. As I mentioned, this is the fourth webinar. We did have three webinars before, one about consumer acceptance, and then the two about one about potato, one the microalgae that I mentioned, and today the toolbox. We have recorded these webinars and we are planning to release the videos later as well, but towards the end of the project. So just a final comment here. So in this sense here, we're talking about using the NGTs, new genomic techniques, like as gene editing to develop organisms with traits that are beneficial to the environment, for the farmers, for the bioeconomy, and the consumers, right? For the uptake, many things to make it work, many things need to be in place here. And so the project is also putting this technology and the study of this technology into its societal context by analyzing different regulatory options, by looking into data requirements for risk assessment, studying the economic impact, and and then public and stakeholder perceptions. I believe that is all I have. And with that, thank you. I invite all to follow LinkedIn and our social media and project. LinkedIn GeneBEcon EU project. You can find us. And if we do and when we do release videos about these webinars and the works that we've been doing, they will be announced in through our LinkedIn and webpage. Okay. With that, I invite Catherine Van Leer to talk about the comprehensive and versatile gene editing toolbox. I think I butchered your name. Sorry, Catherine, but please.
Yeah. Thank you, Igor. My name is indeed Katrijn van Laar. It's a real Flemish name, so I understand for non-Flemish people it's difficult to pronunciate. So I will tell you today about the comprehensive and versatile gene editing toolbox. We call it GeneBWise, which was developed during the GeneBEcon project. And actually, it's a collaboration between my institute, the Flanders Research Institute of Agriculture, Fisheries and Food, but also with a lot of contributions of SPI, who developed the website, and also INRAE and the Swedish University that all produced protocols that were added to the toolbox. So it was a very efficient collaboration. So I will tell you a bit more about why and what the toolbox is. I will then go deeper into the plasmid database that we developed, and then also in the GeneBeWise toolbox itself, so the gene editing workflow and instructions for scientists, and then few things to remember. So let's start with the what and why. Um, so as Igor also mentioned, so within the GeneBEcon project, we wanted to advance and use gene editing tools in potato and microalgae. So potato as an established major staple crop, which is an open field agricultural crop and which has already from literature a lot of established protocols that could be used within the project. And we wanted to improve the starch quality and the resistance towards PVY as two traits. So we want to stack these two traits into potato. Then on the other hand, we had the microalgae, which is a growing sector with a very high potential. And in the opposite of an open field crop as potato, the microalgae are a contained production system. Also for chlorella, one of the microalgae that we wanted to use in GeneBEcon, there are not so many established protocols, even no for some species. So there we had to start from scratch to develop the protocols ourselves. And we wanted to have protocols for gene editing to enhance the production of high value compounds, for instance, the carotenoids. But yeah, CRISPR-Cas, it's often used, but still it's a very complex process. So there are different protocols you can use. You can use plasmid or ribonucleoproteins. You can have agrobacterium transfection, protoplast transfection using polyethylene glycol, transfection by electroporation, by biolistics, etc. So a very complex landscape actually of protocols to perform your gene editing experiment. So for that, it would be very helpful for researchers to have user-friendly and validated protocols, and preferably all in one place, so the landscape of the protocols is not so difficult anymore. And especially when you want to work with complex genomes as potato, which is a tetraploid, or if you want to work with crops that don't have genomic resources and protocols available as the chlorella and the nanofluoropsis, which is microalgae. We also wanted to do multiplex CRISPR-Cas and target multi-gene families. So also that's a very challenging strategy of gene editing. So having user-friendly and validated protocols and workflows for different CRISPR applications would be very helpful. And also for on and off mutation screening, it would be very helpful to have these protocols and different applications. There, I mean to do gene knockouts, to have sequence insertions or replacement, or short sequence insertions or replacements. So these are main applications of CRISPR-Cas. And we wanted to have all the protocols in one place and a publicly available place. So that's what we did. So we created an up-to-date gene editing toolbox. We named it GeneBeWise. And it's developed as a website. So if you go to the website of GeneBEcon, you will find on the right, upper right, you will find a button that leads you to the GeneBeWise toolbox. So it's a collection of state-of-the-art protocols, workflows, and bioinformatic pipelines. It's built as a decision tree approach, and it's meant for high precision gene editing with minimizing unwanted alterations to the original genome, so minimizing the off-target effects. And it's meant for research and breeding purposes, for fundamental and applied research, and for proof of concept studies. And all the the plasmid actually that were used within the GeneBEcon project, we have also collected these on a plasmid database. It's a public repository of plasmids. It's called the Vector Vault database. Here you can see the website. So by this website, this link, you can reach our Vector Vault plasmid database. It's actually a database from the VIB in Belgium, where we had a collaboration with, and that collaboration leads to the addition of our GeneBEcon plasmids to this vector fold repository. And we have a special page dedicated to the GeneBEcon plasmids. It's a collection of 111 plasmids there, grouped according to entry vector, destination vector, or expression vector. So as said, there is a collection of 111 plasmids with a detailed description, as you can see here on the slide. These vectors, the plasmids, they can be purchased through this website, and you can use it by signing an MTA stating that it's only for research purposes and not for commercial purposes. All vectors that we used are based on international standard cloning systems, so it's compatible with most of the experiments. And also the plasmids sequences are added in a universal GeneBank format. Both the sequence and annotation information is also downloadable. And we have included vectors for non-homologous end joining, for base editing, prime editing, and homology-directed repair. Then I come to the GeneBWise, the gene editing workflow and instructions for scientists. If you go to the website and open it, then it's this is how it looks like. So it's built up in 4 panels, from target gene identification to mutants in 4 panels. The first panel is about identification of the target genes. The second panel is on guide RNA design. Then we have a panel 3 on transfection and regeneration. And panel 4 is then on the screening of mutants. And it's built as a decision tree. So by answering a series of questions, the user goes through the whole workflow of CRISPR-Cas. And in the GeneBEcon, in the GeneBEwise toolbox, protocols for potato and microalgae are included, but some of these tools are also validated in other crops, as in Physicometrium and Sichor. So first, the panel 1 about identification of target genes. There you will find a step-by-step approach to identify candidate target genes by gathering detailed information about the target gene, as sequence and annotation, the genomic position, the gene family memberships, and so on, and also identifying sequences with sequence similarity to the target gene. And this is information that is needed in panel 2 for guide RNA design, which minimizes off-target effects. So the GeneBEwise is built through questions. So one of the first questions is if you have identified the target gene in one or more well-studied, closely related model organisms. There you can answer yes or no, because it is important here to go from a model organism to the target organism, sometimes with in-between steps. So how you do this step from model to target is also a workflow that is described there. Another question is to take into account the target gene family. So the question is then, do you have a sequence list of the target genes and the gene family in the model organism? As you can see here, a phylogenetic tree. So this includes the gene in your model organism, but also in other model organisms with sequence similarity, to have the full picture of your target gene related to the target gene family in this model organism. And this is actually the central scheme in panel 1 for the identification of target genes. So you can see how we go from model organism to target organism, which are the requirements, how to generate reference genome sequences for target organisms, how to identify pseudogenes, and so on, how to build a phylogenetic tree of your target gene. So this is all explained, and all protocols are available there. And there's Isham 1 second delay when I click. So it's very important to also have reference genome sequence of your target organism. This is something that we encountered in chlorella. So we designed primers and guide RNAs on the chlorella genome, which is available in NCBI for amplicon sequencing on chlorella strain in our lab. And what we could see is when we designed these primers and guide RNAs on the chlorella genome in NCBI, and then use these primers on our chlorella strain in the lab, we could see only partial amplification or no amplification, or amplification on other places than expected. So it was really important for us to have the genome reference of the genome sequence of our chlorella strain in the lab. So this is the same gene of chlorella, we designed new primers on the strain that we have in the lab, and then we could see that we have really specific amplification, that we have more read depth, and so on. So it was really important to have the genome sequence of our strain in the lab, and that's also something which is included in the GeneBeWise toolbox. And then, as I said, some of these tools are validated in other crops. Here is an example of the tools in panel 1, which we validated in Cichorium intybus, which is industrial chicory. It's a crop that's very locally interesting. We have a long breeding history in my institute in Cichorium. It's mainly used for inulin. The taproot contains a lot of inulin, which is a prebiotic fiber. But also this root is very bitter because of the sesquiterpene lactons. So what we want to do is to minimize or reduce the sesquiterpene lacton content. Because now when you want to extract inulin, you need to do a lot of extraction process to reduce the bitterness, which is a process that is not very eco-friendly. So if you could make roots or chicory roots that are not bitter anymore, then the processing of inulin would be more efficient. So we try to reduce these sesquiterpene lactons. There are already some steps in the pathway that are known. These genes that I indicated here, GAS, GAU, COS, KLS genes, these are known. So these were actually our target genes for Cichorium studies in my institute. So we wanted to have a lot of information on the target genes to design proper guide RNAs. So we had a full annotated genome available of the experimental material that we wanted to use in our experiments. We had the known genes, as I said. We had the GAS, the GAU, the COS, and the KLS genes. And then we wanted to add some more information by doing RNA-seq experiment after jasmonate treatment, as we know that methyl jasmonate induces the pathway of the production of the sesquiterpene lactons. And then we integrated the genomic data that we have, the transcriptomic data we have, to study these genes and gene families in detail. We constructed a phylogenetic tree where we enabled us to identify gene clades. Here you can see also the genetic tree with the RNA-seq data also included. And then for some genes, the genes that of interested, based on the gene plates, we analyzed catalytic activity by a heterologous expression assay in tobacco. And by this, we could select candidate genes for multiplex gene editing. So this is actually the workflow that we used for Sicorium, which is a kind of a validation of the workflow that we described in the GeneBeWise toolbox. Then also in panel one is included the allelic variation. So we have included protocols to measure ploidy level as one of the questions. Do you know the ploidy level of your experimental material? And from this ploidy level, we can then go to allelic variation and also the tools to identify the allelic variation and allelic sequences in the experimental plant material. These tools are also described. So how to see SNPs between different alleles, for instance, as a picture in the slide here on potato. So we have potato as a tetraploid. So we have included the tools to the bioinformatic pipeline to actually identify this allelic variation among the alleles. And then as a final step in panel one, we give tools to identify gene-specific regions because these regions will be important in panel two to design guide RNAs for. And then as an output for the first panel, we have three key outcomes actually. So we have a list of all allelic variant sequences of the target genes, a list of potential off-target sequences, so sequences with high similarity to the target gene, and an identification of unique gene regions in the target alleles, which are then the regions where you will design guide RNAs. And then we go to panel two to actually design the guide RNAs depending on the chosen strategy. And we give in this panel a lot of guide RNA design software. So the first question that we ask there is if you want to have a predefined mutation or a non-defined mutation. With a predefined mutation, we really mean, for instance, when you also include a template so that you really know the outcome, not only the place where the mutation will occur, but also the exact outcome of the mutation. While in the non-defined mutations, you know the place, of course, you know where the Cas enzyme makes a cut. But the outcome, if it's a deletion and insertion, and how many nucleotides involved, this outcome is not defined. So this is the distinction we make in this panel. And so yeah, here is an example of the approach we used in GeneBEcon for potato. So there we have actually a mix between predefined and non-defined mutations. So in potato, we want to increase the quality of the starch by doing actually a two-round CRISPR-Cas. So we have the genes, the GBSS gene, which is involved in amylose production. This gene we want to have a knockout, so the non-homologous end joining by non-defined mutations. We want to knock out this gene to not produce amylose anymore. And then for the other, the amylopectin, so starch is a mixture of amylose and amylopectin. So the amylose we want to get rid of, and the amylopectin, there we want to change the chain length. So we want to have higher amylopectin short change. And for that, we need to have the genes involved SSS1, SSS2, and SSS3. Where we want to have a knockout in the SSS2 and the SSS3 genes. And for the SSS1 gene, we want to insert a stronger promoter there. Yeah, this. And then for the PVY resistant, I think Jean-Luc will perhaps explain it also in his lecture after mine. There we want to have a knockout in 3 of the 4 alleles in potato, and the one allele, the one left, we want to have prime editing to really have a specific mutation which make potato resistant to PVY. So here also we have a mixture between non-homologous end joining, so the non-defined mutation, and then the predefined mutation induced by the prime editing. So in potato, we have this the both strategies in one. Once you have clicked, for instance, on the predefined mutations, then you will have the choice between homology-directed repair, base editing, and prime editing. When you have non-defined mutations, we have the tools for the and homology end joining, and also base editing. And then we will also have the tools to design a single guide for a single target, or multiple guides for multiple targets, or one single guide to target multiple genes, as is explained in this small figure. So here you can see 4 alleles with some sequence variation, and the number 1 indicates one guide for one target, so only for one allele, but also we can have one guide to target 4 alleles, or multiple guides to target multiple alleles. And for each of these strategies, or each choices you make, you will find kind of a table with bioinformatic pipelines and websites and so on, with the parameters for each of these programs to design very specific guide RNAs with minimized off-target mutations. And this is very important, as you see here in this table. So in green is indicated our designed guide RNA for potato, one of the guide RNAs that we designed. But all the others sequences are potential off-target mutations designed by the CRISPR program, allowing up to 4 mismatches. So you can see in the column with mismatches how many mismatches all these sequences have. So you can see for our one guide indicated in green that for this specific guide RNA, we can distinguish some off-target sequences. So it's important to know that for this specific guide, for instance, we have a lot of off-targets. For other guides, the situation was much better, that not so many off-target mutations, or even for some guides, we didn't find off-target sites. So by these programs, you can figure that out, how specific your guide RNA is. So we give all the instructions and parameters, etc., are indicated to do so. And this is again a kind of validation in Sigorium. So also for Sigorium, we use these programs to design multiple guide RNAs for our target genes. And we used Genius and our developed SMAP software package that we designed at ILVO, but which is also based on FlashFry, etc. So we could also design guide RNAs to minimize off targets. Then we come to panel 3. In panel 3, it's about transfection and regeneration with user-friendly and ready-to-use protocols, especially for regeneration. Sometimes it's very important to have some really specific tips and tricks to make it work, as this is a bottleneck for many crops. So also here, protocols and tips and tricks are shared. We have it structured as a transfection matrix. So all the words indicated in bold, if you click on these in the website, you will find protocols, for instance, for polyethylene glycol mediated transfection, for electroporation, for biolistics, for co-cultivation with agrobacterium, for RMP assembly, for construction of plasmid DNA, to isolate protoplasts and isolate single cells. For all these approaches, protocols are there in the website. And also many of them are validated, among others in Sigorium. What you see here on the slide is the workflow for the Sigorium. So we do protoplast isolation and then transfection using polyethylene glycol. And then the regeneration starts. So you can see some pictures on the division of the protoplasts forming at the end microcalli, then macrocali, which we can induce on a solid medium, where regeneration starts into plantlets, and at the end plants in the greenhouse, which we can screen. So then finally we come to panel 4 about the screening of mutants. When you see now in the website, you will see that panel 4 is still under construction. So this is something we are finishing and creating content for the moment. But it will include tools and methods for screening CRISPR-induced mutations. It will be both PCR-based methods, but also sequencing-based methods. And it will contain bioinformatics software for data analysis, and also to screen for intended mutations, but also to identify unintended mutations. So here, yeah, it's kind of an overview of all the tools that will be included in this panel 4. So we will have protocols for the PCR, the amplification of target regions by droplet PCR, for instance. These protocols are there. But then also protocols for the sequencing base, so protocols Sanger sequencing, next generation sequencing. And also when we have the mutant alleles, to bring them back into the gene context to be able to see the consequence of the mutation on protein level. So, and yeah, a lot of tools are also tried and validated on Sigorium. So you can see the same phylogenetic tree as before, but now with indication of the broad spectrum of mutants that we could create here. So for different genes, we could identify a whole patchwork of mutants going from loss of function mutants, heterozygous mutants, homozygous mutants, and so on. So, and all these are like, analyzed, identified using NGS methods, where we could also see mutation efficiency largely varying among guide RNAs. And we could also have the loss of function mutation in 3 paralogous KLS genes, which really led to a reduction and elimination of the sesquiterpene lactone metabolites. So by this, I come to the end of the presentation of the GeneBeWise toolbox. So what is important for you to remember is the website, of course, where you can find the GeneBeWise. So if you go to the GeneBEcon website and then click on the GeneBeWise icon, then you will find the complete toolbox with instructions and validated protocols for multiplex gene editing. And there will be tools for HDR, base editing, prime editing, and non-homologous end joining. And it's really meant for species-specific design of gene editing experiments. It's tools all in one place, and it's a decision tree to follow logic workflow over all the gene editing steps. Then besides, we also have the GeneBeWise plasmids that are gathered in the Vector Vault repository, containing 111 plasmids and built or constructed according to compatible cloning strategies. The tools in GeneBeWise are mainly focused on microalgae and potato, but I showed you also some validation in chicory, and a lot of these tools are also validated in Physicometrium. So we claim that our GeneBeWise toolbox has a broad applicability. When you go to the website, you will have to register before use, but registration is free. So, and if you have questions or whatever, so here are and it's also in the GeneBeWise website, the people that collaborated and are able to give more information in case you have questions. So by this, I come to the end. Thank you for listening, and please, if you have questions, I'm happy to answer them. Thank you.
Okay. Thank you, Catherine, for your presentation and for showing the toolbox. I see we do have a few questions already, which you can type answers during Jean-Luc's talk as well, but we should have some time for answers at the end of the webinar.
Ja, oké,
So
dan
with
ga
that,
ik.
I invite Jean-Luc to present his presentation, how to use the toolbox in the lab, examples from tomato and Arabidopsis. Yes.
Thank you, Igor. You hear me? It's fine?
Yes, we do.
Good. So thank you very much. Thank you for the invitation. So in the talk after Catherine, I will more or less illustrate how good it would be to have the toolbox because it's mostly work we've done before the GeneBEcon and before the creation of the toolbox. But what I would like to illustrate is through different approaches on resistance to potiviruses, is to illustrate how you need to edit differently target gene to achieve what you want. So just basically, we research center in south of France, we work on resistance to viruses. And the idea is to develop genetic resistances to viruses, so mostly crops from Mediterranean area, but then we think also those mechanisms can be translated to other crops. And the idea of developing resistance for plant breeding is that those resistances shouldn't impair the plant development and they should be of high durability. And we mainly work on susceptibility factors. So susceptibility factors, it means those are the factors that the virus will hijack to infect the plant and so to develop their cycle. So the idea behind that is if the host factor is modified or suppressed, the virus can't use it, and then you will have a genetic resistances. And because those mechanisms are quite well conserved, you can hope you can transfer them to other species. So in the case of GeneBEcon, it will be, for example, potato. And before I go to genome editing, so just to present, we are working on resistance to potiviruses. It's a large group of single-strand RNA viruses. It's very important for many crops, such as potato with PVY. So you discussed that at the latest webinar. Sharka for stone fruit and cucurbits and everything. And for the last 20 years, it has been done that one family of plant genes encoding translation initiation factor are very important for those viruses. And so, as I said, the viruses, potiviruses, they need to hijack mainly EIF4E. You will see it's a protein that is shown linking to the cap of the mRNA, and they use it to perform their cycle. And by looking at this mechanism in different crops, so it would be pepper, tomato, but also the model crop Arabidopsis, the idea is to understand how we can use those factors to design, using genome editing, to design resistance in other crops. So just to start briefly on the origin, originally it was known that there were a lot of natural resistances in pepper in mainly area. Either you could check cultivar cultivated all over the world, or you could sample semi-domesticated plants as here in Mexico on the top right hand. And those resistances, there are more than 30 alleles of resistances that had been isolated. Those resistances were associated with mutation in the protein EI4E in the gene. And in fact, they were not knockout of the gene, but they were amino acid changes. And as you can see on the bottom right, you can see a 3D structure of EIF4E. And most of the mutation amino acid changes associated with resistance were located in two regions, which is 1 and 2. So that will be the yellow loop and the red one. And so the first idea was, what can we do if such resistances have not been selected during domestication? And the obvious idea is if you inactivate the gene, you will get resistance. And for example, we knew that if you inactivate EI4E2, that's one of the gene encoding for translation initiation factor, in big fruit tomato, you would have a resistance to a few isolate of potiviruses. And so if you want to use this mechanism to design resistances, then you will use very basic CRISPR-Cas9 as shown by Catherine, which means you will have a single guide, a Cas9 protein. You will target two regions of the EIF4E. You will see the two targets at the bottom. And by doing so, you will generate insertion or deletion of nucleotide and inactivation of the gene. This you can check. You can check by sequencing, as has been shown before. You can sequence the genomic DNA, but I must recommend it's very good to check also on the mRNA to check that what you see on the genomic is really expressed in the mRNA. And here, as we've done, for example, it's good also to check on protein, and you can see that we got 3 lines, 2A, 2B, 2C, where you can see that there's not any more EI4E2 protein expressed. And those proteins are resistant to a papaver motor virus. So that's the most easy way to generate resistance in another genetic background by using this simple gene knockout. So now what we found when we did that, so you can inactivate different EIF4E in tomato, for example, is that you somehow don't have the same resistance as with natural allele. And the reason for that is because of gene redundancy. And for example, in tomato, you got 2 genes, EI4E1 and EI4E2, that both encode protein involved in translation initiation. So then, as you can see on the successive panel, if you inactivate 4E, most of the viruses will use 4E2, and you will have a very limited resistance spectrum. You can do the reverse, that's just what I show. You inactivate 4E2, you will have most viruses using 4E1, and again, a very limited resistance spectrum. What can work is you can inactivate both EI4E1 and EI4E2, so then you can use, for example, a multi-guide strategy, and you will have a very strong resistance spectrum to a lot of potiviruses isolate. But then, as you can see here on the right of the picture, this is a healthy EI4E1, EI4E2 plant. It's a double knockout, and you can see that the plant is not doing very well, and it's not going to be very good for plant breeding. And the reason for that is that besides the role for virus, translation initiation factor, as initiating protein synthesis, you will understand is very important for the plant. So that will be a limit. And what we know from studying the natural variability is that what's going on in a wide accession, for example, as I said, you don't have gene knockout, but you have gene modification associated with amino acid changes. And so, if you can summarize, what we know from studying natural variation and domestication in tomato, but also pepper, pea, and so on, and is that you have a trade-off between resistance and development of the plant. And because EI4E are both important for the virus, but also for the translation, you need to find a balance between both activity. And in the natural compartment, this has been selected through those natural resistance alleles, which are still functional. So you got resistance, but you still got the functionality. And if you want to knock out genes, you will have resistance, but losing the functionality, and then you can have a lot of problems. You can have developmental defect, you can have a limited resistance spectrum, you can also have increased resistance breaking. And so it's something probably nearly 10 years ago, when genome editing was really becoming something that could happen, we thought if we want to use genome editing, we don't want to do gene knockout, but you want to copy more what has been selected through natural selection. And so it changed completely what we wanted to do. And the first thing we did, so we use Arabidopsis using transgenic plant to check if it works, is we check that you can copy mutation from one species to another. For example, on the left, you can see a natural resistance allele of P to potivirus. And we show that if you just introduce the same modification, so it's going a bit too far away, same modification of the Arabidopsis gene, you could generate resistance without affecting the development of the plant. And it's because those mutations are located, have been selected in a special part of the protein, against region 1 and 2. It doesn't change the structure, it doesn't change the function, but it will change the electrostatic potential at the surface of the protein, which means that in fact, what happens is EI4 still works to initiate translation, but it will just repel the viral protein that wants to interact with it. And so I'm going really quickly because it's a bit out of the topic, but we check, because there was a lot of amino acid changes, we looked at the role of each amino acid independently, and we found that two combinations, that will be the, if you look, the T80D, S81D, and especially the asparagine 176K, which are ticked with a green tick, are sufficient to drive resistance. And again, those are mutations that will change electrostatic potential on the surface of the protein. And this was interesting. So we thought, now, instead of doing gene knockout, we need to do gene modification and really copy mutation that happens. And just to remind you, if you do gene inactivation using CRISPR-Cas9, in fact, you use a guide to guide the Cas9, you generate double-strand break, and then because you got a pyramid repair, you will have insertion or deletion. What we wanted to use was base editing, where the Cas9 doesn't is a nickase, it only breaks one strand of the DNA, and it's associated with a cytidine deaminase, so it will just directly introduce point mutation. So it's a cytidine to T or C2G or C2A. And we are very interested because we had noticed that if we want to introduce asparagine to lysine 176 mutation, we had an ideal location for a sequence guide with a perfect location of the PAM site and everything. So all the things you will find in GeneBecon's website. And we knew that in this case, if we use a single guide that is very specific and Cas9 associated with a cytidine deaminase, we could try to generate this mutation. And very briefly, it's what we did. We construct with N-Cas9 cytidine deaminase and the guide in a Y-type Arabidopsis. And from the first generation, we found a lot of mutation exactly at the site we wanted to target. And in 2 generations, we could generate plant homozygous for N176K mutation. It means it's plant with a very precise mutation at the site we're targeting that will induce amino acid changes in the encoded protein. And very briefly, what we can see is that those proteins are functional. So we can see that the plant has no phenotype when we just modify this amino acid in the protein, but also they are very resistant to a clover yellow vein virus. So we can achieve this time the trade-off between development and resistance. So then we went back to tomato because Arabidopsis was more to have a proof of concept because it was easier to and faster to get. And in tomato, you can see on the top, you got natural resistance from Solanum abrocaetes species accession. It's a white tomato with very small fruit, green fruit that are not very edible. And on the bottom, you got EI4E from the Solanum lycopersicum, that is a full susceptibility virus to the gene allele to the virus. And we didn't know which amino acid was the reason for the resistance, so we choose another tactic. So we decided to mimic what's going on during selection to modify both region 1 and region 2 of the EI4E. For region 1, we used a HDR approach with homology-directed repair. So in fact, because it was quite old, we didn't even use CRISPR, but we used TALEN. But you could, if we had to redo it, we will use CRISPR and a template that will be inserted through HDR in region 1 to insert a mutation. And for region 2, we use a citidine-based editor, so exactly the same system we used previously for Arabidopsis. But we also use to have a faster selection of the mutation. We use a co-editing system with ALS to select directly mutation through resistance to chlorsulfuron. And I'm not going to show you all the details, but in the end, we managed to get at least 15 alleles with modification in EI4E. Which means it's a way of generating a high level of diversity at a single locus in the genome. And of those, we selected here 3 lines, one that is mutated only in region 1 through a knock-in, one with a mutation in region 2, and one with both. And those are the mutations. You can see they are just represented by the little circle, and you can see them on the 3D structure. We could check that those proteins, when modified, are still expressed the same ways as wild type. So during those modifications, we do not change the expression and the stability of the protein. And then we checked for resistance to viruses to a very good isolate of PVY, that's N605, that is quite a stringent isolate for tomato. And what we found was that, you can see here, it's ELISA data that just show the level of accumulation of viral protein in infected tomato. You can see that if you only edit one region of EI4E, you got no resistance. If you edit only region 2, that would be sorry, I can't point, but you got no resistance as well. But if you manage to edit both region 1 and region 2, then you will get a very good level of resistance and very little accumulation of the virus. And this we could also prove using a GFP tagged virus. You can see from left to right, different plants with inoculated of GFP tagged virus. You can see the virus appearing in green. And on the right, you can see that the edited plant for region 1 and 2 is completely resistant to the virus. And what we did after that, we checked also the culture of the tomato. As you can see, we did a greenhouse assay, and we found that we can have those resistances, but with no impact on the plant development. And because the EIF4E mutation are really mimicking natural mutation, you got no effect on the EI4E translation initiation. And so you can manage to get at the same time resistance and plant development. So conclusion, we think this work shows the importance of susceptibility factors to generate genetic resistances. But as I said, you have to be able to balance resistance and development. And this gave rise to a translational project to provide and extend those resistances mechanism to other species. And that would be, for example, potato for resistance of PVY, as presented last week by Laurent, or last time by Laurent Glay. But we also try to carry on similar project on cassava for cassava brown streak virus. And as you see, depending on what you want to achieve, you really have to think about the strategy you want to do. So that's where you will definitely need a decision tree and see which tool you want to take in the toolbox to manage. But as we won't have time maybe to discuss today, but it's part also of things that have been presented by Catherine, gene knockouts can be interesting. Base editing can be okay, but still limited. So prime editing would be the best way to get the mutation we really want to get where we want. Just very briefly, so it has been quite a lot of work and a long work because we didn't have at that time the right tools to generate. But I think with the current advance in genome editing, it would be much easier to get the similar works, which we hope we got very quickly in potato. And I think that will be okay for me. And thank you very much.
Okay, thank you, Jean-Luc, for your presentation. Before we get into questions, we'll have a little bit of time for 15 minutes for questions. So we'll be able to look into the ones that were done. As I mentioned, this is a last webinar in a series that was done for the project GeneBEcon, and it is our plan to publish all of them soon on our website and through our social media. So if you have missed it, hopefully you can still watch all of them. So with that, if we go to questions, I think we have some answers already, but maybe we can comment further for the attendees, Catherine and Jean-Luc. Well, first point is there any possibility to share the slides that I can answer as already done by typing? Yes, we can share. We also have the recordings available, right? So people can watch again. I believe if that's okay with the speakers, we can also put in our website to make it easier to download. Catherine, there's a question about training course. Do you want to comment on the upcoming event you mentioned?
Yeah, yeah, I also already typed something in the chat, but I can say it here also. Yeah, actually, we were not planning within the GeneBEcon project to give trainings or courses on the GeneBWise toolbox. But I'm also member of the AAB Planted group. This is actually, there was a cost action before on plant genome editing. And yeah, this cost action is already finished, but the network is taken over by AAB. It's some British organization that organizes events as conferences, training schools, and so on. And one of the training schools they are doing in August this year in Düsseldorf is a training school on gene editing, where I also invited as a trainer. So of course there I will teach and use in my courses the GeneBWise Toolbox. And I know from this group, the AEB group, there will be more training schools organized in the future. I don't know when or where, but I know they plan to do each year a training school. So these will be occasions to perhaps teach and use the GeneBWise Toolbox in training courses. So yeah, that's for the moment what we are planning to do as teaching and things. But this is not in the framework of the GeneBEcon project.
Okay.
But of course, if there is a lot of demand, yeah, we can see.
Okay. Yeah. Thank you. Another question we have is specifically how can the GeneBEcon toolbox address off-target risks realistically, ethical concerns, scalability beyond model plants while navigating EU regulatory constraints? I believe this question was put just even before we are about to start our presentation today. But beyond what you already typed in, do you want to comment on some of the general aspects?
Oh, no, I think what I typed there is actually my answer. So yeah, we try to do guide RNA design as specific as possible. So that is the main realistic thing to address the off-target risks. And also in the screening, we have the workflow there, how to really screen for these off-target sites if there are mutant alleles there. But this is for the predicted off-target sites, of course. If you want to go overall, then yeah, you need whole genome sequencing. This is feasible from a practical point of view, but not realistic, I think, in terms of regulatory constraints. But in the GeneBeWise toolbox, there will also be a workflow on whole genome sequencing. So it will be there, but if it's realistic in terms of legislation, that's something else. And then, for the scalability, so I hope I already showed in my lecture that the tools we use are transferable to other crops, as I showed in Cichorium, but also in INRAE, they did a lot of things on Physcomitrium, and also Jean-Luc used, I guess, most of these same or similar tools on tomato. So that's about scalability, and the more the GeneBeWise is used, the more scalable it perhaps will be. So so I think that's my answer. I don't know if there are other,
As we
And
have
about
a,
ethical concerns, it's also related to another question on IP that I could see in the questions. So it's not really included in our GeneBeWise toolbox about IP, but we have this ethical claim that it's used for research purposes, not for commercial. Also for the vectors in the repository, users will have to sign an MTA stating that it's for research goals and not for commercial goals.
okay, thank you. Let me address a technical question that just showed there. Someone was trying to register and there seems to have an error message. You should be able to put an email. If you're not registered yet, put an email and a password, click register instead of login. You have a few more like name and information to put in. You should be getting an email for confirming, basically validating the email you provided, and that should give you access directly to the platform. So there's no specific requirements other than having a valid email in the standard email format. If you do have issues still, feel free to send us a message asking. It can be to communication@GeneBEcon.eu, and we should be able to check what is going on. Okay. So we do have a new question. So it was mentioned that there was the need to develop KOs in both E1 and E2 in tomato to get complete resistance because of redundancy. However, when you introduce these specific changes, you can reach almost complete resistance by focusing only on one of the proteins. Can you explain this?
Yes, I can try. I mean, I can't explain it, but I can. It's a very good question. It's something we're working on, and it's also one of the important way of using changes, amino acid changes, rather than knockout. Is as you notice, if you knock out one gene, the other, the 4A2, for example, will just take the place and be recruited by the virus. But if you modify it, if you just repress the interaction with the virus, then it's functioning on its own. So we are working on different hypotheses. One of the things we think is that because the EI4 doesn't work on its own, but within a translation complex with EI4G and plenty of protein, as you saw on the first slide, there must be competition within those complexes. So which means that if you modify EI4E1, it will be in the complex, and then it will just bar the 4E2 from being recruited. So it's one of the things we are working on, and we would like to understand in the future. So thank you for the question.
Okay, thank you. We have one more question. Dear professors, can we integrate machine learning within gene editing technology to make progress and more robust and accurate? I guess it goes to you both if you have seen this or any discussions in that sense.
Yeah, it's a difficult question. It would be nice. We are in my institute also thinking on how to integrate machine learning with gene editing technology, and we are now thinking about project proposal on how to use machine learning to predict outcome of mutations. So yeah, that's one way how we think machine learning could be integrated with gene editing. But yeah, I don't think we are already there. I don't know, Jean-Luc, if you have more things to add
No,
on that question.
no, I think I agree. It would be very interesting. I think especially it would be interesting in the for prime editing. So prime editing, it's a bit different technique. It's more complicated. It's a method that could allow us to do the mutation we want and turn it into a mutation, another mutation, because you just introduce a template that is replacing the original sequence. And it's very limited in efficiency. And probably it has been done for animal cells and other systems. It would be good to know exactly how to design those guide RNA to do that. But the problem maybe with machine learning in this case is that you need to have a lot of data to train your machine learning. And so you need a model where you can use probably design a lot of guide and train them. So maybe Fiscomitrella could be a good model for that. But for sure, tomato won't be, because you can't make thousands and thousands of prime editing in tomato. So probably protoplast cell studies can be something, I don't know. So in our hands, we're not going to do it, but it will be probably interesting.
Okay, thank you. Yeah, I think these are all the questions. I do see the technical question was already problem solved, so I'm glad to hear. And very good. Okay, I think I don't see any more questions. We still have 5 minutes. So if anyone has a final question, it is the time to do that. But I don't see any. Okay. Catherine and Jean-Luc, thank you for your presentations and for the answers. Do you have anything else before we close the session?
No,
No,
thank you
no.
for the
I
invitation.
hope everyone here uses the GeneBeWise toolbox. So
Okay, well, thank you for all the attendees, and I hope the presentation was, I believe it was good information, and as Catherine mentioned.
Igor, I see hands raised, or I don't know.
Oh, I think they're just claps. They're just,
Okay, sorry.
I guess it's a thank you since they cannot put in the chat a message. So I thank
True.
all for attending, and as I said, follow us on LinkedIn, check our website. We'll be promoting the videos that were recorded and the outputs from the project. So, and the GeneBeWise is available through the website as well. So do check it. Okay, thank you, and see you next time.
Thank you. Bye bye.
Thank you.
Bye.